And almost all of you have already had to practice that. So hopefully you'll be excited with what has come up today. So with that, I'd like to jump right in and tell you what I've been working on these past several years in Chicago, but also the Pacific Northwest. So depicted here is this incredible high diversity band of life that occurs along those lines in this temperate rock intertidal system. My lab has worked on for many years. So this is a high productivity, high biomass zone of life. It's a narrow zone of life with strong species interactions occurring within just a couple hundred meters from shore. Due to high light availability in this shallow zone in the marine system, as well as elevated nutrient concentrations, which can lead to strong and interesting ecological changes. All right, so I'm going to jump right into this with a brief case study that highlights the importance of nutrient effects in community structuring. So this is one of these ecological changes that can happen in the coastal environment. So in a natural experiment, a group of researchers from California looked at some Aleutian islands off of the coast of Alaska that are fox invaded as a result of the fur trade and another set of islands in this Aleutian island archipelago that were not invaded by foxes. And so here's some typical Aleutian islands. Marine fish preyed upon by seabirds. Excretion from these seabirds delivers a rich marine nutrient source to those islands in a shuttle that promotes plant growth. Infestation by foxes in this system has removed the native seabird prey. This nutrient shuttle is completely cut off, and the islands, as a result, are transformed to this plant-depauperate tundra. So the native grasslands are completely transformed just by the loss of this nutrient resource. That works this morning. This is like totally not where I want to be standing. Okay, so the temperate rocky shore on the U.S. West Coast, which is specked with this diverse and abundant life, is characterized by high productivity and a characteristic animal signature in nutrient chemistry. So ammonia, the form of nitrogen that's excreted by animals, is elevated immediately adjacent to the coast. So here I show a plot of mean ammonium concentrations in monthly water samples taken at repeated transect off of Tatoosh Island, heading out into the seawater, not close enough to land to get a signal from that. And you see a really strong ammonium concentration onshore that dissipates almost completely by 1,000 meters offshore. This is seeing a green light. It knows that it's trying. Did it work? I don't think so. Take that. Okay, so coastal marine systems are typically nitrogen limited, and so this animal ammonium signature onshore actually is a strong driver of productivity. So I'm going to show here 3 small case studies that were really instrumental in driving my ideas in developing this thesis. The first, a paper from Kathy in 2007, showing a long-term experiment that she has run. This is over the course of a few years, where she's gone out and repeatedly removed mussels from a series of tide pools and left mussels in place in another series of tide pools. Here's a picture of mussels in a tide pool, for those of you who aren't as familiar with the system. And she found over and over that pools with mussels present have higher ammonium than pools without. In any scenario, pools have more ammonium than the ocean. So that's that gray line at the bottom there. And interestingly, this is the paper that I was reading at Oregon State in 2008 when I was developing my master's thesis. Thought this was a really cool little result, and my master's advisor said, oh, that was my roommate once upon a time. You should probably keep reading about her stuff. So thank you to Sally for funding this paper. So a related paper, Matt Bracken, around the same time, was looking at similar questions in tide pools, and he found that this ammonium that is elevated in tide pools, so again, that red line in mussel-present tide pools, is correlated with increased algal growth in those same pools, as opposed to pools without mussels do not have that translation into algal growth. And then finally, a third study, this study looking higher when there are no algae attached to that barnacle. So these white specks here are barnacles. If you picture just a small red alga attached to that barnacle, you can actually measure less ammonia getting into the water column if there's an algae attached to it. So this is a really tight link, and it's a really important driver of productivity in this system. So our understanding of microbiomes living in association with macrobiota in coastal systems is a rapidly expanding field of study, as Kathy started to mention. So as a product of a coastal nitrogen working group in Massachusetts almost 2 years ago now, some co-authors and I, mostly shown in this photo here, have put together a review paper that suggests that these associations between macrofauna and macroflora, so these large-bodied plants and animals, large-bodied in our system being things like seaweeds, maybe not large-bodied for most of us, showing that nitrogen cycling is actually augmented by this presence of a diverse microbiome in association with these macrobiota. So here are some of the macrobiota that we looked at in this paper. So fish associated with remineralization, mussels with nitrification, the sponge Anomox, another sponge with nitrification, alga, nitrogen assimilation, and seagrass nitrogen fixation. These are all microbially driven nitrogen metabolisms that are enhanced at these nodes of macrobiota. So this suggests that incorporating microbial communities into our picture and our analysis of this deeply studied coastal marine system has the potential to really increase the number of nitrogen-based interactions that we might expect in this coastal system. So in the nitrogen-limited coastal intertidal system, the dominant paradigm for coastal productivity is that oceanic upwelling delivers nitrate to the nearshore intertidal zones, which constitutes the major quantitative nitrogen pool available to marine algae and angiosperms like seagrasses. However, the nitrogen regeneration pathway, or excretion of animal-generated ammonium, actually provides what could be a preferred nitrogen source for marine algae. It also is available to marine ammonium oxidizers. So these microbial communities are capable of converting ammonium to nitrite and nitrate via the process of nitrification. And so what I've spent my last few years thinking really carefully about is how is it that rocky intertidal algae and microbes might be actually interacting in terms of this shared nutrient resource, animal-regenerated ammonium. So this red arrow here has been my curiosity for the last several years, and I'm going to tell you how I've thought about that in these next half an hour or so. So I've addressed this one question, I've told this one broad story using 3 separate pathways, and I'm going to outline them here. So the first pathway, on a small scale, I performed a series of mesocosm experiments to consider the algal-microbial interaction in terms of ammonium in a highly controlled system. I then went into the field to consider the environmental drivers of microbial community structure. Given my understanding of this interaction in a really controlled way, it was of interest to see how applicable is this in the real life. So I have a vested interest in a community ecologist and not looking at tanks for 6 years. I'd rather be in places like this. So that was where I moved on to as soon as I could my PhD. And then finally, I had a curiosity of blending these first 2 chapters together. So given what interaction I could measure and what environmental drivers I could see in the field, how important is an interaction that I could measure in the lab in a manipulative experiment in the field. Okay, so my first chapter took place at Friday Harbor Labs, shown here. This is an unfiltered image. This is what Friday Harbor Labs looks like from the bluff above the lab. There's Mount Baker in the background. This is a small town with a fabulous marine lab located just northwest of Seattle. You get to it by a series of ferries and cars. It's a really exciting place to work, and I felt really fortunate to spend a few years there. And this is where I performed my mesocosm experiments. So in this chapter, I'm going to refer to a whole slew of response variables. So I'm going to run through a quick primer of what icons I'm going to use to talk about these response variables. So seawater nutrient concentrations, or DIN, the dissolved inorganic nitrogen that I can measure in seawater, is high surface area substrate for microbial community to settle upon. So this is a replicated way for me to sample the microbial community in these systems. I measured algae on two different scales, two body sizes. I looked at microalgae in the periphyton. So pictured here is actually a box of microscope slides that was submerged. It provides a standard surface area from which I can scrape that microalgae. Called a periphytometer. I invented it. The macroalga that did all right in tanks was Prionitis. This is a large-bodied intertidal algae common throughout this system. And then finally, as a proxy for primary productivity, I measured chlorophyll A concentration by extracting samples from that periphyton. Okay, so to investigate this interaction, I was interested in thinking about how this interaction could be occurring in two directions. So here's my highly simplified system that I brought into the problem, thinking about ammonia as a shared resource for microbial biofilm and algae. My first question was, how is it that algae are responding to ammonium availability in these tanks? So following up on what has been shown over and over in tide pools, can I make something like this happen in tanks? And then to loop across to the other interactor in the system, how does algal abundance drive microbial metabolism of the nitrogen resource? And then in the other direction, how is it that microbes are responding to my experimentally elevated ammonium? And how does microbial abundance drive algal uptake of that nitrogen resource? So in thinking about this first direction, the influence of algae on microbial nitrogen use, I performed a 2x2 factorial experiment with 2 levels of ammonium and 2 levels of algae. The 2 levels of ammonium will be depicted in the lighter hues versus the darker hues. And the 2 levels of algae, when I have ambient periphyton in this system, is the green hues, and reduced periphyton will be the gray hues, the white and the darker gray. So here's what that looks like at the lab. So the shaded tanks is how I reduced my periphyton load. The high periphyton was just subject to ambient sunlight. So by adding plywood, I didn't change the temperature of the system because it's a flow-through seawater system, but I did completely eliminate the periphyton concentration in those tanks, and I'll show you some data to support that right now. So here is what my first result from this mesoplasm system is that algal growth seemed to be pretty much independent from ammonium availability in the ways that I was looking at it here. So on the first y-axis here, chlorophyll A concentration is the circles. The dry mass on that right-hand vertical axis is the dry mass scraped off of those slides. And you don't need to know which shape is which because they follow the exact same trend. That I had higher periphyton in my higher light treatment with no response to ammonium availability seemingly. So here's what that looked like. So if I took the plywood off, that's what it looks like in the bottom. On the top, this is disgusting on purpose that when I have ambient light, this is the bloom that can occur in a tank. And it's a really highly productive, highly photosynthesizing community of diatoms primarily. So the second set of responses that I'll show here are seawater nitrogen concentrations, or DIN, the dissolved inorganic nitrogen. These are all going to be shown as change from ambient concentration as a result of my algal and my ammonium treatments, with the solid horizontal black line indicating no change. So that's the 0 line. So the first step here is the resource that I added. I was mimicking animal-excreted ammonium. Ammonium was added experimentally, and this resource was drawn down by algae. So the 2 lighter hues are at the 0 line. When I don't add ammonium, it is not increased over the course of an experiment. But the 2 darker hues on the right are elevated above that 0 line, with the green hue being lower than the gray hue. So the algae, when I have ambient algae, the ammonium is drawn down. So the second result is nitrite. So I'm showing these in the pathway that we would expect if nitrification were occurring. It's the order in which I'm presenting these, the suite of nutrient measurements that I made. So if nitrification were occurring, that ammonium is converted by the microbial community into nitrite. And here's what I measured in the system. Nitrite was indeed drawn down by the algae. And that drawdown was less severe when ammonium is elevated. So if you compare those 2 green hues, the far left and the finally, nitrate, the third product of this ammonium oxidation process. Um, Nitrification and ammonium oxidation I sometimes use interchangeably. Those are the same process. Ammonium oxidation is a little bit more of a technical term telling you what is going on. So nitrate, the second product, which is also, remember, the most abundant form ambient in seawater, was also drawn down by algae, so those two green hues below the zero line. But this drawdown was less severe when ammonium was elevated. So if you compare those two green hues, that nitrate is higher under the high ammonium. So possibly suggestive of microbial production of nitrate under the high ammonium conditions. But everything you see here is a snapshot measurement of standing nutrient concentrations. So to obtain some mechanistic conclusions about nitrification, I applied a heavy nitrogen tracer to this system. I applied heavy ammonium, so an elevated 15 net signature as opposed to 14. Added it as ammonium, measured the nitrite pool to see how much of the ammonium I added was actually making it into what I suspect is the first step in nitrification. And here is a summary of the results from that quick experiment. And here I found a reduced microbial nitrification rate in the presence of algae, but only when ammonium was elevated. But I didn't see this reduction in nitrification rate under the ambient ammonium conditions. So before I walk you through my first set of microbial community results, I'll step back a minute to walk you through my approach to collecting microbial community data from the mesoplasm system. So the bioballs, before they ever made it into the mesoplasms, were put in a big mesh sack and actually trucked off the dock at Friday Harbor for a couple of weeks to be seeded with the ambient local microbial community. They were then distributed among these treatments randomly and collected samples as bioballs, extracted DNA out at Argonne National Labs, thanks to Jack Gilbert's hospitality. And then these DNA samples were run through a high-throughput sequencer to get data on the V4 region of the 16S rRNA gene, and what comes back is a slew of genetic sequences that I have learned to align with known taxonomy, known microbial taxonomy, and perform some techniques in community structure analysis that I'm going to show you here. This is the only pie chart you'll see in this plot, but it's the easiest way to, here's a community. Okay, so here is the first microbial result I'm going to show. This is a PCOA ordination showing that microbial community, this is plotting microbial communities based on phylogenetic similarities. This is based on unifract distance. So the closer together 2 points are, the more similar and the more closely related the communities in those samples were. So I found what looks like an interactive effect of periphyton and ammonium. So high periphyton, these green hues, were distinct from the shaded, these white and gray hues at the bottom. The taxa correlated most strongly with this distinction, so OTUs or operational taxonomic units were all photoheterotrophs, so Dinoroseobacter and other from the family Rhodobacteriaceae, which are strongly dependent on light availability to exist. So that makes sense. Clustering by ammonium treatment, though, only occurred under that highlight condition, that I didn't see an ammonium effect unless it was in that higher light with the increased algal presence. So that's where you see the distinction between the two greens, the dark on the left and the lighter on the right. And the taxon that seemed most strongly related with this separation was an archaeal genus, Nitrosophilus. The Latin name Nitrosophilus maritimus translates to tiny ammonium oxidizer of the sea. This guy is actually capable of utilizing ammonium at a quicker rate than phytoplankton in all the systems that it's in. So this is a relatively new finding, but a very exciting little guy. Okay, so with that, considering the other interactions, so we know that the microbial community was influenced by this ammonium. How is this interaction going in the other direction? So here, again, I used a 2x2 factorial experiment, elevating ammonium at 2 levels, ambient and elevated, and then I made an attempt at changing the microbial community, elevated versus ambient. Elevated meaning that I put those seeded bioballs into the tanks and let them stay put for the whole experiment. I reduced that ammonium, the microbial load by swapping out those bioballs almost daily during the experiment. So starting that successional process over and over to reduce what biofilm was existing in my 4.5 liter tanks. when microbes are elevated, they're pulling down that ammonium relative to that reduced microbial content. So nitrite, what we suspect to be the first product, if nitrification is occurring, was produced under high ammonium conditions. So indicating the presence of a nitrifying biofilm, whether I elevated the microbial community or not, that anytime I added ammonium, I see this increase in nitrite. And then translating up to nitrate, I see drawdown across the board. But this drawdown was, again, less severe under elevated ammonium conditions. So the two darker hues on the right are drawn down less. Again, indicative that nitrification could be going, that these mesocosms with seeded bivalves, especially, so if you look at the difference between the blue hues in, I think I should be pointing at this, the blue hues in the left-hand pair, that lower pair of nitrate points, and the dark hues on the right-hand pair of nitrate points. The blue hues are always taller, closer to 0. So last drawdown when I have a lower load of microbes. So I used stable isotope analysis to measure nitrogen use by the macroalga in these mesocosms, which is Prionatus. So at the end of the experimental period, I found no difference in algal percent N by any of these treatments. But when I looked at the isotopic ratio in those same plants, so when I took the alga out at the end of the experiment, dried it, ground it up, I see a decrease in del 15N. This is the ratio of heavy to light nitrogen in the tissue. So light isotopic signature, less 15 to 14 in the treated tissue, comes actually as an artifact of my experimental treatment, that this industrially produced ammonium chloride is isotopically light. And so I can make an inference that the nitrogen being taken up by the plants in the course of this experiment is indeed the nitrogen that I added to the system. That no change in percent N shows that any nitrogen being used is probably being used for growth right away. There's not a standing stop building of that nitrogen, but the source that they're using for that growth is the one that I added to the system. So chlorophyll A concentration extracted from the microalgae in these tubs was elevated under high ammonium conditions, but at both levels of ammonium, so the 2 points on the left and the 2 points at the right, I see a muting of this response by the elevated microbes. So those brown points are lower than the blue points, showing that the presence of these higher microbes hinder the amount of productivity we can measure from the algae. So to conclude this chapter, here again is my simple system, and the quick answers to my questions. Number 1, algal growth seemed to be relatively unaffected by ammonium availability in this little system, but that abundant algae muted microbial utilization of ammonium when that ammonium was elevated. In the other direction, microbial nitrification indeed increased in the presence of elevated ammonium, and these elevated microbes seem to mute algal utilization of ammonium, but only when it was elevated. And so what I have concluded from all this is that I do measure competition for this ammonium resource, which illuminates the importance of animal-excreted nitrogen or animal-excreted nutrients in mediating what could actually be going on in this system. So I have several slides like this added in, that when you've done a lot of hard thinking in the field science, it's nice to look up from what you're working on and look across the street and see the Olympics, for example. So I've done the same for you in this talk. Let's take a break. We're going to leave the mesocosms and move along to the second chapter. So once I had an understanding of the potential range of algal-microbial interactions in terms of nitrogen availability, I was really interested in seeing how important these could be in an unmanipulated environment. I also had a vested interest in, like I said, leaving these tanks and going places like this. So the algal-microbial competition in terms of ammonium in the mesocosms was pretty clear, but what is driving that microbial community structure and distribution across a pretty patchy ecosystem? The topography is variable, the light is variable, the wave action is variable, and it covers a lot of real estate. So here I performed a nested biogeochemistry spewed across the San Juan Islands, from which I can make a lot of really interesting inference. So here's what this looked like in real life. Tide pools were compared with adjacent emergent rock. So on the left here is a bioball pouch bolted onto the rock in a tide pool. On the right is its pair on emergent rock. And so as I showed earlier, we know that pools with their associated invertebrates are able to ameliorate nitrogen limitation that can occur at low tide when all of these plants on the exposed rock are isolated from their nutrient resource, which is the ocean. Pools are inherently different places to live than the adjacent emergent rock, a pattern which I've shown here for macrobiota, so invertebrates and algae in this system. So in this ordination, I'm displaying the macroalgae survey data that I collected concurrent with these microbial samplings, showing, again, samples closer together are more similar in community composition. And here, emergent and pool samples cluster really neatly across NMBS axis 1, regardless of site. And interestingly, about 70% of the taxa driving emergentness in this ordination are algae, whereas in the other direction, I have a really nice mirror trend where about 70% of the taxa driving a positive score on NMBS axis 1 are animal. So here are the PCOA ordinations plotting microbial community samples in the same comparison, emergent versus exposed rock. Here, emergent rock is red and blue is tide pool. These are ordinations based on unifrac phylogenetic similarity again, so based on how closely related the taxa in these samples were. And in both study years, pools in blue and emergent rock in red, communities clustered separately, whether I considered the relative abundance of taxa, so these weighted unifrac, or just presence absence, so unweighted unifrac. So let's take that a step further and see who is actually driving that. So here are bar charts from those same samples, in which each bar is a sample from 1 year on one site, and each color represents a single OTU, so a taxon from the microbial data that I received. So echoing my previous slide, the most visually striking pattern, which the color maybe is better on this TV monitor, is this difference between habitats, with sites and years actually looking remarkably similar. And indeed, I didn't get a significant site effect. So the word you can't read at the bottom is sort of on purpose, that those groups of 5 bars are different sites, and yet the pattern seems robust across those sites and between years. The rows there are years. So despite what appeared to be similar differentiation between habitats and between years, I found actually strongly significant clustering between sample years. So here, I'm plotting those same points, but this time coding for year. So pools and emergent rock are separated here and colored by red as 2013, blue as 2014. Again, weighted and unweighted phylogenetic similarity. So clustering was significant using both of these distance measures, with a stronger clustering when taxa were only considered as presence absence. So this bottom row, the unweighted unifrac distance, which is hinting at the fact that rare taxa with low relative abundance are actually really strong players in driving this community clustering. So I wanted to push that a little bit further. When I ordinate samples from these 2 years using a distance measure that considers each OTU as a completely unique individual, doesn't take into account the phylogenetic similarities, and here they're ordinated using Gray-Curtis distance, I found even stronger differentiation. So this indicates that the taxa driving this significant clustering were actually locally closely related. So when I take away information about their phylogeny and count them all as unique, they could not be more different on this ordination across PC axis 1 year. So to further investigate the importance of rarity in driving the between-year difference in these samples, I performed a series of PCOA ordinations with increasingly stringent sample membership requirement. So what do I mean by that? Here I'm plotting OTUs that occur in at least 5 of the 40 samples in pools and 5 of the 40 emergent rock samples. When I punched at this cutoff, considering OTUs occurring at least 15, 25, and 35 of the 40 samples from each habitat type, I found that clustering actually collapsed at that most stringent requirement level. variation is robust between years. So in this survey of microbial community composition across these 4 sites, I found a strong habitat signal, a weak site signal, and a strong annual signal that was explained by these rare, closely related taxa. So from this chapter, I can conclude that microbial assemblages are driven by habitat-associated parameters. So what I showed here is a summary ordination displaying just 1 year of microbial data. This is the 2014 data, coded again by habitat. So the blue is pool habitat, the red points are emergent rock habitat. And significant environmental drivers of this pattern that I collected simultaneously are overlaid in a joint biplot. So anything significantly correlated with this ordination pops up as a gray arrow. The parameters explaining my pool samples, so that significant factor in the ellipse there on the right, are indeed descriptive of the pool environment. So high ammonium, high phosphate, high animal, so this is an animal metric, it's the amount of animals per total things living in that spot. While the parameters explaining the emergent rock on the left side of NDS axis 1 are descriptive of the emergent habitat. So higher light, high temperature fluctuation, high DO, or the dissolved oxygen measurable at that emergent spot. So not only were the differences in microbial community between these habitats and just who was there, but I also have some hints at differences in functionality. So here I'm plotting the mean number of OTUs in this data containing gene sequences that we know to be associated with nitrogen fixation. So I see a really clear elevation of this signal on emergent rock compared to pools at all 4 sites. And the taxa really influential in driving this pool emergent rock difference in plots like this are typically cyanobacteria. So from Pseudomonas and Xenococcaceae show up over and over as incredibly strongly correlated on emergent rock, almost non-existent in the tide pool samples. And so we see this clear elevation in a habitat where the only available nitrogen for a huge part of the day is N2. So this is not a biologically available resource. This is something that has to be fixed before it is used. And so even though this is not a measurement of actual nitrogen fixation, it's an indicator that the capacity for nitrogen fixation could be there more in my emergent rock samples. And so the big conclusion from this chapter is that the pool emergent rock habitat distinction is strong, and it is driven by these related environmental parameters that are pushing microbial community composition and function in the field at all 4 of these sites. Okay, so with that, I'd like to walk you quickly through my last chapter here, which is the consideration of this algal-microbial response to artificially elevated ammonium in situ, in real life, out in a place where these things actually live. So my question driving this last chapter was, is ammonium alone a strong driver of community structure and function? So not just the microbes. How strongly is ammonium driving this system? So my mesocosms highlighted the potential for algal-microbial competition for the shared resource. My observational survey illuminated the potential importance of high ammonium in tide pools specifically as a microbial community driver. So how are these gills responding to ammonium in nature? So I'm going to start with where I chose to do this study. I chose 2 study sites from that observational suite of sites that differed strongly in macrobiotic community composition. So here's an NMDS ordination of macrobiota at the 2 sites I selected. These were collected from vertical transects, that's what that white tape is there. I ran a transect line from the ocean's edge up to the top of the intertidal zone, wrote down what was attached to the rock every 25 centimeters, all the way up, it could be 1,200 meters up the coast, up the shore there. And I did 16 of these vertical transects across each site, so spanning about 100 meters, to give me sort of a wiggly grid of who is inhabiting each of these 2 sites that I was looking at. So the plotted data here are the results of these vertical transects. The sites cluster separately along axis 1, with the taxa driving my cattle point samples being almost entirely animal, my green line doesn't change, the purple line seems to increase over the course of this 4-hour assay. This is repeated one time, but a replicated experiment over and over, I see an increase in nitrate adjacent to bioballs that had been exposed to this higher animal community structure. So a quick indicator that maybe these sites are interesting microbially. So this together with the dissolved oxygen drawdown in those purple points there, this is an oxygen required process of converting ammonium into nitrate. So suggests that the microbial biofilm at Cattle Point has a pre-existing condition of being capable of metabolizing ammonium better than the microbial community at Westside Preserve. So with that motivation, I deployed ammonium dispensers in the intertidal zone at each of these 2 sites. Here's what they look like. So on the top there, this is the base of the dispenser. It's a PVC cap and a PVC plug that have been perforated with a drill press. The cap stays put attached to the rock all summer. The plug is filled with agar in which I've dissolved ammonium chloride. I've perforated the edge of that plug. And so once that agar has set, it's a solid block of agar full up of nutrients, and I can screw it into that cap. And I can replace that with fresh nutrients every 14 days or so over the course of the summer to have a pressed condition at these sites. I repeated this 8 times across each of these 2 sites and had adjacent control spots that were far enough away from these dispensers to not get spillover effects. All at the same tidal height. So these were all underwater for about the same amount of time at these 2 sites and exposed about the same amount of time. I had a suite of response tools clustered around each of these ammonium dispensers, these little experimental cities. So there's the ammonium dispenser in real life. I have a spot where I installed dissolution blocks. This is a gypsum or calcium sulfate block put into the rock. I did this for 4 24-hour periods over the whole course of the summer to get a baseline for what the water motion is at that site. The amount that the plaster dissolves, I can get information about how much the water is moving at that site. Microalgae were measured. Right next to the dispenser there is a microscope slide that I've epoxied onto the rock that I can swab for chlorophyll extracts. Some bare rock right next to the dispenser inside that same red circle. I used a razor blade to scrape microalgae occurring in a biofilm right on the rock there. And then again, our good friend the bioball was installed in Vexar pouches immediately adjacent to the dispenser that I could subsample from that Vexar pouch in this repeated high surface area. And so what did I find? First of all, comfortingly, when I added ammonium at both of these sites, I indeed created pool-like ammonium conditions that occurred at both sites. So here on the x-axis are my 2 sites, the y-axis is the ammonium concentration that we measured in the field directly above those dispensers, and at the same time scooped from the emergent rock, scooped from the tide pools at that same site. And by adding ammonium, I create the yellow bar looks a lot like the blue bar at both sites. I see higher experimental ammonium delivery at Cattle Point, which I infer to be a result of higher water motion. So those plaster blocks showed higher water motion at Cattle Point, which could be driving this increase, even though the amount of ammonium I was adding on day 1 at both sites was the same. The amount of water washing over it right away when I came to take my sample was higher at Cattle Point, which could explain that big yellow bar there. But by elevating ammonium on emergent rock to make them more pool-like, I actually was able to create a microbial community on the left here, the Westside Preserve. They're colored the same way. Blue is pool, gray is emergent rock, yellow is my dispenser. I created a microbial community that actually fell pretty neatly between emergent rock and tide pool communities in this PCOA organization at Westside Preserve along this axis 1, but I saw no such clustering at Cattle Point, where ammonium concentrations in pools is already quite high. And so I suspect that my treatment didn't maybe create truly pool-like conditions at Cattle Point, as the water motion prevented that ammonium from being sustained at these high levels. It could also have been affecting it to other parts of the system. But in the site where ammonium is lower to begin with in pools, and water motion is lower, I was indeed able to create a microbial community that approaches a pool-like community. The primary producers in this system seem to respond to my ammonium treatment unevenly. acting differently between the two sites. So on the bottom here is what chlorophyll A looked like across the 30 summer sample months, June, July, and August in 2014. And on the top is the west side preserve. The triangle is my ammonium treatment, the NH4. And at west side preserve only, I see a chlorophyll effect that develops over the course of the summer that did not occur at cattle point. So indicative of the ammonium being more important to primary productivity, more of a potential limiting resource at that site based on this data set. So a quick summary of this experiment is that the use of ammonium by the benthic community is actually pretty complex, is all the hints that I've gotten from that quick analysis so far in this data. So I did indeed elevate the ammonium to pool levels at both sites. I saw uneven water motion between sites, which could explain, be partly explanatory of why my ammonium drove microbial community to be more pool-like at my low animal, low flow site only. The microalgae showed a small productivity increase following my ammonium treatment, but again, this was only seen at my lower animal, lower water motion site. So, back to my question that I began this talk with, how is it that these rocky intertidal algae and microbes are interacting in terms of this shared nutrient resource? My first chapter, I hope convinced you that algae and microbes have the potential to compete for ammonium, but only when it's abundantly available in my mesoplasma system. Although the distribution of microbial taxa was broadly regional, but my sites didn't differ, the microbial community structure was actually strongly environmentally selected. That microscale difference was really important to driving this turnover. Ammonium availability seemed to be a stronger driver of ecological responses in general at my site where it's more likely to be a limiting resource. So I see this context dependence of the ammonium resource between my two sites that I chose to study. And so let's come back to the review paper, because I think this is a really nice way to come back and think about this project in a bigger context. So we, as part of this effort, spent a lot of time thinking about microbial participation and community interactions as a really important contribution to ecological understanding of a lot of these long-studied coastal systems. And as part of this paper, we put forward a series of hypotheses that could push forward our understanding of microbial function in the coastal ocean, where nitrogen is typically a limiting resource. And the two of those that I want to mention here, that I think I have addressed through this study, is that one, microbiota, macrobiota associations seem to broaden the nitrogen cycling function that we could expect to measure in these coastal systems. And then second, these associations are likely to produce some ecological links that have not yet been considered. So these novel links among macrobiota are actually facilitated by microbial nitrogen cycling function. And this is a really exciting way to push this field forward in a place that we know a lot about the interactions already going on. And so that is all I have. I have a series of acknowledgments that I'll go through quickly, and then we can get right into questions and let you all get onto your holiday weekend. But I want to start by thanking really sincerely my committee. First of all, for being here on your literal last day on campus before you leave for the summer. Come back from Hong Kong, you're amazing.
Just for you, just for you.
I'm really fortunate to have all of you in the same room. I really appreciate it. Kathy has been an incredible advisor in her confidence that I actually went to Friday Harvard to do a 6-week experiment my first summer, and she's like, then you'll come back to Katush and we'll get going and we'll do more stuff. And I never came back. I just thought, Kathy was like, well, it's not working at all. Like, everything you're doing is not great. But I kept pushing, and Kathy's perseverance pushed me to, I actually, there is something there, and if you keep working, and this has sort of been what I repeat to my field health workers when they're like, this is terrible. I'm like, well, it's something. And that's sort of a really inspiring thing. So thank you, Kathy, for being confident enough to let me do that. The rest of my committee has provided a suite of just deep familiarity with this system, deep understanding of ecological analysis, and enthusiasm for micro I did not come in as a microbial ecologist. I still hesitate to use that. Like, I talked to microbes for an hour, but thank you for being brave enough to let me do this and invite me to your lab so warmly, Jack. I also want to take a step back and thank my master's advisors who really got me started in this field, took me from the coast of Maine to the coast of Oregon, and I never want to leave again, that the Pacific Northwest coast is a really special place. And Sally remains one of my closest advisors and closest colleagues. We're still working on papers together, and really pushed me to stay in this field, and I really am grateful to these two for that. I want to thank my welcome into a system that has been studied for so long. So this is something that I found my last field season at one of my sites. It's like right next to one of my biolog badges. I don't know whose it is, but this is a transect that has been studied, transect A, FHL 1971. So this system really is deeply studied, deeply understood, and the fact that there are still new questions is really exciting. And so I want to thank the history of my system. I also want to thank University of Washington Friday Harbor Labs, the BLM, and the San Juan County Land Bank for really generous access to all the sites that I looked at here. I was fortunate to be funded really well throughout this process, especially the Friday Harbor community is really warmly accepting of non-University of Washington students, that I'm a Chicago student that was treated equal to the UW students in terms of financing and resources and colleagues, and it's really been, I feel really fortunate to have had that access. I want to thank my technical support that helped me do some of these really technical approaches that I had no idea what I was doing on, and really got me through some of these isotope analyses and microbial sequence analyses at both here and at Argonne. My support team in the E&E and the VSD and the VSCD, in terms of all the logistics here, the grad student support from Allison, Libby, and Audrey has been both friendship and logistics, and hugs and tears, and you're okay and you're not okay. They've gotten a lot of us through a lot of hard times, and we're just eternally grateful to that trio. Eric Larson in the VSCD has been an incredibly inspiring teacher. That this is someone who tells the story behind biology and really gets students on board with that, both non-majors and majors. And I really credit him with all the teaching experience I've gotten here and being able to continue on with it. I want to thank my lab. This is a phenomenal group of people that have really, we support each other and we've really gotten through a lot. Special shout out to Amy, who was my first in Chicago and at Friday Harbor. Courtney, who came to my house for a 30 practice yesterday. Our first years who fled the state before but really brought a lot of enthusiasm, and like Courtney said, we saw what it was like to be a first year again. This was a really great thing. They also, these poor guys, were present for 3 6th years finishing, which is a horrible time to do it. Through. I want to thank the Darwinian cluster in general, our colleagues, grad student peers, a really amazing group of people. I've once shown this cohort picture. I wanted to show this cohort picture. This is what we looked like when we got here. We all looked fresher. My field helpers, I had a lot of help in the field. I want to thank them because they could have turned around and stopped counting algae and seen orcas. Instead, they did something like this for me. And they're out their backs to the ocean in this picture. And I really got a lot of amazing data as a result. My FHL group, so a lot of people I think are tuning in on the computer next to John there. Like I said, this was a community outside of Chicago that I consider almost equal to my community here. The friendships and the amount of science that we got done as a group was really amazing, and I thank them for welcoming me into their group. And then my family. So these are my parents and my brother. Turns out we take a lot more pictures, either when we're getting married or having a baby, so every picture of my family is one of those two things. My parents are from Maine. They brought us to the coast early and often, and they really inspired this love of being outside, and I thank them for that. My other family here, I all of a sudden got 5 siblings when I married John, so this is representative of this giant family that has been endlessly supportive of me as well. in the intertidal ladder way. So that was a good sign. This guy has, we moved to Chicago together. This has been an amazing adventure, and I wouldn't get through the day-to-day without having you by my side, and I'm really grateful for you getting me to this day in particular. He last weekend took the baby to a White Sox game so I could write my talk. That was incredibly great move. These three, my first children, Mark, Kim, Melanie, and Kyla. And then, of course, this guy, Paul Krobowski, in his thesis defense, referred to Casey and Louie as the important result of his dissertation. And I was like, so cute. But, like, that's so true. Absolutely, what has driven me to really be inspired to keep going these last several months. And if you have noticed, he may be back in a few years. This is him wearing the Broomball team shirt. Thanks for doing that. And so, with that, thank you for your time, and I'll take any questions.
I'm really interested in this divergent microbial communities between the emergent habitat and the pools, and considering that they're not exposed all the time. My assumption is that the environmental conditions are very similar with some water stuff, but maybe you have a better sense of whether there are conditions that persist even when the tide is in.
So it's certainly a completely different community. So when I measure the macrobiota in pools and not pools, they're really different. They fell apart really neatly on that ordination. And so who is there is different. So the nutrient drivers are going to be really different between those two places. But like you said, the wave action is so strong that could be swamping that signal completely. And I don't know the answer to that. My hunch is that the deep disturbance that happens with that tide, that complete desiccation, the high exposure to light, the huge temperature fluctuations, is probably enough to reset that community in a way that's completely different than happens in the pools. And that can't be overcome by being underwater for part of the time, but I don't know the answer to that. It's a really interesting temporal thing to look at.
Following up, were there other, the two biggest drivers in the microbial communities between those habitats seem to be the nitrification factors. Were there any other microbe traits that you looked at that are drivers?
So I looked at using this, the proxy for thinking about the presence of different genes that we know are participants in microbial metabolism. So nitrification and nitrogen assimilation, denitrification, nitrate assimilation, is that the right word? Distillatory nitrate reduction. Yeah. I do know this word. So we looked at that. So that is not a measure of function. It's a measure of genes that we would expect to maybe be involved in the function. It is not a perfect tool, but it can give some hints at it. And I showed you the most strong result there. The nitrogen fixation difference was the strongest result I found using that tool. But we do see some hints of changes in the dissimilatory and assimilatory nitrate reduction based on this ammonium treatment. But I haven't pushed that boundary yet. But I have the data to do it, so that could be. I'm looking for projects for my undergrads next year, so I have a lot of data, so things like this could be.
And cyanobacteria are big.
And the cyanobacteria are a huge distinction. Yeah. Right. So yeah, we can search the table for just names of things we know are participating in these processes. And so the biggest difference I'm seeing is nitrifiers and cyanos, but there's 47,000 in taxa there, so I could keep reading.
So in the last project where you made the, just by adding ammonium on the emergent rocks, the biofilms got more like pool-like communities. Does that mean that all these microbial taxa are just like ready to go? Like, because you saw totally different taxa, so they like zoomed into that area, or they're just all hanging out in the ocean at any moment?
I think the understanding is that they are. This is a big debate, right? I think that they're ready to go. That the function it's been referred to as microbial seed bank. So that's one way to think about these taxa that jump in and are ready to fill that role. There's also